Okay, so, okay, good. We've got closed captions. Gonna move this. Thing's kind of bulky. We're gonna get that out of here. All right, here we go. All right, tissues, chapter 5 in your lab manual. Earlier, we discussed cells. Generally, cells are the smallest unit of life. So when we discuss levels of organization, that's kind of where we start. Now, chemistry is kind of associated with that cellular level. So when you watch the video from this morning, you'll hear me discuss carbs, carbohydrates, proteins, and fats, and how the different cellular organelles work with these items. So anyway, ultimately though, we're going to put these cells together and we're going to end up with tissues. So as we go into tissues, we will see some cellular information and we will discuss cells a little bit more. But yeah, tissues are what tissues are made up of cells. And then when we put tissues together, we end up with organs. Okay, so this is kind of our next level of organization. Okay, so a bunch of cells put together give us tissues. When we do like a biopsy, we're analyzing the cells, but we actually are taking a tissue sample. Right? You look at a tissue sample, you see multiple cells. Okay? It's very difficult to just extract one cell and just look at a cell. So we look at a tissue sample to see a multitude of cells. And we know from this what tissue and cells are supposed to look like versus what disease or potential cancer may look like. All right, so tissues. So we're going to see there are 4 primary tissue types. So we're going to spend a little bit of time discussing each of those tissue types. The first one is epithelial. We'll probably spend the most time on epithelial tissue. Epi, you may recall, as a prefix means upon something. Epi means upon. Connective is next, and then muscle, and then nervous. So I'll go into details about each one of those as we go. Now, it gives you some basics regarding function up there. For epithelial tissue, it's primarily going to be a covering or a lining. A covering or a lining. So any organ that has a covering or a lining, that's going to be comprised of epithelial tissue. So one covering that we see all the time is your integumentary, right? Your skin and your hair. Okay? That is epithelial. What we see is the epidermis specifically. The type of tissue is epithelial tissue. So then we'll look at some of the specific cells associated with the epithelium of the integumentary system. In a little bit. That's just a fancy way of saying we're going to look at skin cells. Skin cells of the epidermis. Okay? So epithelium is a covering or a lining. So the skin or the epidermis of the skin would be a covering. A lining would be like the inside of your nasal cavity or the inside of your mouth, inside the lining of your digestive tract, the lining of your urinary tract, the lining of your reproductive tract, the lining of your blood vessels. If it's a tube in the body, it's got a lining. And that lining is made of epithelium. Okay? Some epithelium is going to have specialized cells that secrete mucus. Our mouths have a little bit of mucus, right? Our nasal cavities, our digestive tracts. So we'll look at some secretions that oftentimes come with epithelial tissue. So a covering or a lining. So what functionality-wise, that would probably offer some protection, right? So we think of one of the functional aspects of epithelium is that it does protect. Not always. We'll see different types of epithelium that don't protect, but primarily we think of that as protection. Also offers, in essence, a barrier of some sort, which kind of goes with protection. Connective tissue is just deep to epithelial tissue. So underneath the epithelium, or deep to the epithelium, you're going to find what's called connective tissue. So going back to the skin, the epidermis is the covering made of epithelium. What's deep to the epidermis, or what's under the epidermis? It's called the dermis. The dermis. So the dermis would be connective tissue, for example. Connective tissue is just that. It connects, it links the epithelial covering or lining with the underlying soft tissue, which is typically muscle, our third tissue type. So these kind of go in order of superficial to deep. Epithelium is always the most superficial. Connective is always below or deep to epithelium. It connects the covering with the underlying tissue, which is muscle. Skeletal, so we'll see that. 3 types of muscle that we're going to look at. The most common, of course, is the skeletal muscle. So going back to the skin, we've got the epidermis on top, or the epithelium. You've got the dermis in the middle, the connective layer. Then we'll see another kind of connective layer of fat or adipose. And then that's going to take us to skeletal muscle. Nervous, well, and then in the guts, we'll see what's called smooth muscle. So not just the guts, the entire viscera, your cardiovascular, your blood vessels, your urinary tract, your reproductive tract, your respiratory tract, your digestive tract, all those tracts are lined with epithelium, and they have a little bit of connective deep to that, and then a bunch of smooth muscle. Smooth muscle is going to be involuntary. We don't have control over it, whereas skeletal muscle is voluntary. You have control over it. Look at a few of the differences there. Then, of course, we have the cardiac muscle as well. And again, we'll see the inside of the heart, the chambers of the heart have a lining, an epithelial lining. And they have a little bit of connective tissue underneath, and then the thick, what they call the myocardium, or the muscle of the heart. Again, epithelium on top, connective in the middle, muscle on the bottom. And then nerve or nervous tissue is kind of, we call it ubiquitous, or it's kind of everywhere. Nerve tissue is all over the place. Except epithelium, we don't really find nerve tissue within epithelial tissue. Generally, epithelium has no blood supply and no nerve supply. So nervous tissue is going to be kind of interspersed or mixed with connective and muscle. And of course, nervous tissue is the electricity that provides nerves and a pathway to deliver messages to the brain and then back out to the body. So basically, in order to get the muscle to contract, we have to have it plugged in. So the nervous system is plugged right into all these muscles. We also find nerve and blood within connective layers as well. So we're going to describe each of these, go into a little more detail as we go. Okay. So gross versus histology. Gross anatomy is what we're going to be doing next week and really throughout the semester. Gross anatomy is what we could see with the naked eye. They also call it macroscopic versus microscopic. Macro means large scale. Okay, so macro or gross anatomy. Now, an image would be considered gross anatomy. If I brought in some X-rays or MRIs, those are considered gross anatomy. I can see anatomy with an image with the naked eye. I don't need any type of scope. Okay. So then microscopy requires some sort of viewing lens. So histo says histology equals microanatomy. More specifically, really, histo deals with tissues. So histology is the study of tissues. Cytology would be the study of cells, right? Cyto means cells. But generally, we discuss viewing microscopic anatomy as being histology. Because again, we don't really look at individual cells, we look at groups of cells, which are tissues. All right, so let's start with epithelium and work our way through. Again, functionality-wise, it's a protecting, has some protective qualities to it. It is a covering or a lining. We do see some filtration going on, particularly in the kidneys. A little bit in the lungs as well. We have to transfer oxygen in to the bloodstream and pull carbon dioxide out. So we'll see kind of a filtering of chemistry there. We'll see in the guts, especially the small intestines, absorption of nutrients. So again, the lining of your guts is going to be epithelium. The thing about epithelial tissue is it's very hypermitotic. That means it goes through cell division quite rapidly. Like your skin cells, your epidermal cells, they only live about 28 to 35 days. It's pretty rapid. So every month or so, you've got a whole new batch of epidermis. In fact, most dust and dander in your house is dead skin cells that have sloughed off over time. Like your vacuum cleaner. A lot of that dust in your vacuum cleaner, believe it or not, is dead skin cells. Same holds true with the guts. High percentage of your poop is lining of your intestines because it's constantly sloughing off or leaving. Okay. Okay, absorption of nutrients. Sensory input, we do, again, there's not really nerve directly linked or directly within epithelial tissue. However, that underlying connective tissue is full of nerve. So we have what are called nerve endings that basically plug into the basement of the epithelium. So the epithelium can still sense temperature, right? We can sense temperature. I can sense pressure. I can sense touch. I can sense certain things with my skin, but it's not because I have nerves in the epidermis. It's because I have nerves plugged into the underside of the epidermis, and they can receive some sensory. Secretions and excretions, I mentioned that a little bit ago, but yeah, we have to lubricate a lot of our tubes. Some characteristics of epithelium. Again, generally we'd see these in tightly packed groups, especially if we're providing protection. So like the epidermis is a bunch of tightly packed groups of cells. And again, these aren't slam dunks, right? These aren't always situations. This first bullet is these are possibilities. Okay. Each cell is polar. That means it has poles, like a north pole and a south pole. It has a top and a bottom. Apical means the top or the superficial aspect of the cell. Apical is like the word apex. kind of the top of the cell. Basal means base. So the deep part of the cell would be called the base or the basal surface. And then the top and the bottom. Apical is the top, basal is the bottom, and then around the sides, we're going to see surface connections occurring. So cells are going to be connected to one another. And we'll look at a couple different types of cellular connections. Cells are avascular. So A, remember, means not or without. Vascular has to do with having blood vessels. So avascular means without blood vessels, without a blood supply. So how do we feed? Don't cells need to eat? Why do we even have a blood supply? Why do we have blood? To deliver nutrients, right? To take away waste. Got to bring the groceries to the cells, and we got to get rid of the garbage. The blood is what's doing that. The plasma is carrying all the nutrients. Red blood cells are carrying the oxygen. How do we feed the cells then? They don't have a blood supply. Well, again, the basement or the base layer is connected with connective tissue, which is full of blood. So we can get nutrients into the extracellular space of the connective tissue, and they'll feed up, diffuse, typically to the basement of the epithelium, which makes sense because that's where the connective tissue is. So the basement of epithelium is primarily where we see the new cells. That's like the nursery. That's where mitosis is occurring. And then as these cells grow and age, they actually move closer to the surface. And they're basically dehydrated. So at the base layer, you have these nice, puffy, hydrated, brand new baby cells being fed from underneath. And then the next day, a new cell is born. So then this original, it gets pushed up a little bit. And the next day, a new one comes in and it gets pushed up. By a certain amount of time, that original baby down here, after a few weeks, is now dehydrated, is at the surface, and is getting new nutrients. So it becomes a dead epithelial cell. And it does what we call sloughs off. Again, that's what most of what's coming off your skin. Old keratin cells. Innervated. So again, innervated means nerves plug in. Okay, so again, the nerves aren't necessarily coursing through the epithelium, but nerves do innervate, again, more or less the basement, because the connective tissue is where all the action is. Connective tissue is full of blood supply, full of nerve supply, full of lymphatic vessels. Okay. So again, we can kind of plug in nerves from the connective tissue into the basement of the epithelium. And it allows for some sensory aspects to take place. Okay. The cell membranes of all these cells are connected with one another. We call them junctions. Okay. So where these cells join are called junctions. And we have different types of cellular junctions. And again, we'll look at a couple of illustrations of these. We have what are called tight junctions. Now, some cells, depending again on the organ, some cells of the organ, they strictly are there to kind of create some sort of boundary. And so they might have more or less tight junctions. They don't necessarily need to communicate with one another. They don't necessarily need to pass material between each other. So they're just basically there as like cement. They're there for support and structure. So those are where we find tight junctions. So, and again, I'll pull up an illustration. Desmosomes, there's a little bit more of a connection or link between the two cells, the neighboring cells. And then gap junctions, they allow for transfer of fluids, nutrients. Okay, so again, I've got slides for each of those. And then the basement membrane, just like it sounds, that's going to be a membrane or a kind of like a wall that's going to connect the base layer of the epithelium with the connective tissue. So where connective tissue and epithelium connect, that's called the basement membrane. Remember, the deep part of epithelium is called basal or base. So the basement layer is the deep layer that brings the connective tissue together with the epithelial tissue. Okay, so tight junctions, when we look at those, again, those provide a lot of structural support. It's kind of like brick and mortar type stuff. A brick and mortar, that's what tight junctions are. No leaks. We're going to fuse cell membranes together. So all these neighboring cells are fused together, forming tight junctions. And again, these allow for strength and durability, as well as support. Desmosomes also provide quite a bit of strength and support. They connect the plasma membranes as well of adjacent cells, but a desmosome provides a little more flexibility than a tight junction. So we're kind of going in order of most stable or strong to weakest. So strongest is the tight junction. That would make sense. Desmosomes next, still pretty tight, but we do have some extra little filaments that allow for some flexibility. So again, certain tissues get a lot more action than other tissues. Okay. So repeated tension and stretching, okay. That sounds like fascia, which is going to be surrounding muscle tissue, kind of skeletal muscle. Okay, so desmosomes. Okay, and then gap junctions, the third type, those are, I guess you'd say maybe the weakest of the links, but what their goal is to provide some sort of link or connection with neighboring cells. So we can actually have a tight junction or a desmosome and still have a little bit of a gap junction somewhere connecting or the neighboring cells. Again, this is going to allow for transfer of cytoplasm, transfer of or cytosol, maybe the liquid inside the cell. We can potentially share nutrients. So, so maybe one of these cells is a lot closer to the action for nutrient absorption, so it absorbs nutrients, and then it can transfer those and share with neighboring cells via these gap junctions. And again, remember, these are membranes combining with membranes. And one aspect of cell membranes is that we have what are called integral membrane proteins. In other words, we have little proteins kind of embedded in the membrane of a cell. I also call them channels. These integral membrane proteins form channels. So a lot of times when these cell membranes connect, we can link channels together. So again, we can transfer nutrients or fluids in between. Maybe lysosomes or peroxisomes, which are your immune system aspects of a cell. So if a neighboring cell is more infected with a potential pathogen, then a different cell can maybe send in some troops, some peroxisomes or other immune factors that can enable the other cell to kick out whatever's there. And then lastly, like I mentioned, the basement membrane. Anytime you see basal or basal, it just means basement. And again, that's going to kind of cement or glue typically the epithelium with the connective tissue. It says cement cells to underlying tissue. And again, most of the time that underlying tissue is going to be connective tissue. Okay, so when we get to the skin here shortly, we'll notice where the epidermis ends and the connective tissue of the dermis begins. Okay, we're going to notice a distinct kind of basement membrane. Okay. So the basement membrane isn't really part of the epithelium. It isn't really part of the connective tissue. It's more or less kind of a foundation or a divider or a kind of a wall of protection. It keeps the epithelial adhered to the connective tissue. Okay, so the basement allows for adherence of epithelium and connective. All right, and then lastly, for epithelium, or not lastly, but the last little batch here, and then we're going to classify some of the specific epithelial tissue types. The surface of an epithelium or an epithelial tissue is, again, dependent on where it's located. We see some surfaces that are relatively smooth. We'll see others that have what are called microvilli or a brush border. One thing about structure of organs, especially more or less on the tissue level, we do see folding in a lot of places. And what folds do, folds provide more surface area. So if I want to go, so like these microvilli, for instance, if this surface was flat, if I go from here to here, it's a flat surface, so I have nutrients coming through the lumen of the tube, and they're supposed to be absorbing down into here. If this is flat from here to here, it looks like about 2 feet. If I fold these, if I put a fold here and a fold here and a fold here and do that this whole way, now I can create a whole bunch of feet of surface area. You guys see that? I mean, I can get this much surface area in just one curve. So villi and microvilli provide surface area. They maximize surface area for absorption. We want to absorb as much nutrient as we can in the guts. So the microvilli are generally found in the small intestines. Again, all of this information we see it throughout the semester. So you're getting just that kind of intro foundation. So when we see it each week, we don't have to reinvent the wheel. Okay. And then cilia are a little bit taller, a little bit longer than microvilli. Okay. By the way, too, this is not, maybe cart before the horse, but I do want to make a side note, FYI, for now, put it in your back pocket. The microvilli, these little fold, or the kind of the area, like with my microvilli, there's blood supply that goes up into it. But that blood supply is still coming from down here, from connective tissue. So this curve or fold is still going to have a basement. So the basement membrane will continue up and back down, and then you'd have a blood supply because these microvilli are absorbing nutrients. So we need the nutrients going into the blood. There's no blood supply here. So I have to kind of punch up a blood supply in through that microvilli. So anyway, keep that in mind. You'll see it in a lot more detail when we get to digestion. The cilia, they're not for absorption. Cilia are generally found in the respiratory tract and therefore filtration. They kind of move particles around. They move mucus around. So if you got mucus build up and it's coming up from the lungs, it's the cilia of the lining of the respiratory tract that are kind of moving all that goop around. Cilia generally are a little bit sticky, so they can trap like microparticles. So when you're breathing, and you breathe in dust or dander or other people's skin, these cilia, again, they're kind of sticky. So they'll those microparticles will attach, and then the cilia will, through wave-like motions, will kind of bring that gunk to the outside of the body. Okay, and then the last one, keratin, that's what we see with the epidermis. So when we look at the skin, we'll see keratin as one of the specialized surface cells. These are, it's a form of protein, and keratin is going to be hard. It's kind of like dead skin cells have become keratinized. So remember when I said a skin cell or a lot of cells are born in the basement and then they kind of dehydrate as they move up? Well, they're becoming keratinized as they move up. So as a cell, epithelial cell dies, it becomes dehydrated and therefore becomes keratinized or kind of hardened. So keratinized kind of means like hard, like dehydrated, dead skin cell or dead epithelial cell. So like when you get a pap smear, what they're doing is they're looking at the cervical epithelial cells. They want to make sure the cervical epithelial cells are not keratinized. They should have some mucusy, they should have fluidity to them. They shouldn't be dehydrated and hard. If they are, that means that there's a possibility that we're in a hypermitotic stage. It's kind of like a callus. Like if you do this over and over and over, you shovel or whatever, that constant friction builds up calluses and you get hyperkeratinization. And it's because the skin cells are hypermitotic. They're saying instead of lasting, a month, they might only last a week because I'm rubbing them off so quick. So then I have to build more cells faster. So if I have a keratinized cervix, well, if I have a keratinized cervix, I'm in big trouble. But if you have a keratinized cervix, hopefully it's nothing too major, and it's just simply like a dehydration. Maybe we've got some imbalances with some specific minerals, or we're lacking some healthy enzymes. But dehydration can lead to keratinization where it shouldn't. Okay, so epithelium. So how do we classify epithelium? A couple different ways, primarily based on how many layers and what shape the cells are. Okay, so the number of layers of cells it's either one layer or it's more than one layer. Okay, if it's one layer of epithelial cells, we say that the layer is simple. Simple means one layer. Stratified simply then means more than one layer. Simple is one layer, stratified is more than that. Multiple layers. Simple is single, stratified is multiple. We primarily are going, so if epithelium, one of its main functions is protection, we're probably not going to see simple or one layer for protection. We would see multiple layers. So a lot of our epithelium is probably going to be stratified. Just throwing that out. It's stratified. Most epithelium is stratified. We do have a third one that's not on there. If you put the prefix pseudo, which is spelled P-S-E-U-D-O, pseudo. You heard that? You've seen that, like a pseudonym, or means like appears to be but isn't, maybe fake, pseudo. We sometimes put that in front of stratified. So we'll have pseudo stratified epithelium. We only really find that, I believe, in the respiratory tract. It appears to be stratified, but it isn't. And it's columnar epithelium, which I'll get to in a second. All right, so anyway, this is a side note. You'll see that. These three are the shapes of the cell. So we not only give it a how thick is it or how many layers adjective, we also give it a what is the shape of the cell adjective. Okay, so squamous, cuboidal, and columnar are the options. Squamous means squashed or flat. You can see squamous going here is flat. And then, you know, what we do is we put one of these in front of squamous. So it's either simple squamous or stratified squamous. Simple squamous would be one flat layer of epithelium. You guys see that? Simple, one, squamous, flat. So one flat layer. That's definitely not going to give anybody any protection. Okay? Simple squamous is for gas exchange, rapid exchange of gases. So we would find that in the alveoli of the lungs. Again, if you just simple squamous, maybe air sacs or something of the lung. We do, again, we do respiratory, you'll hear all of this again. And as it relates specifically to each system. Okay. Again, if I need to get oxygen in, carbon dioxide out. It's got to happen quick. Okay. I don't need a bunch of thick layers to go through to get oxygen in and carbon dioxide out. So that's where we find simple squamous, pretty deep in the lungs. Okay. Let's see, stratified squamous, so many layers of flat tissues. We find that in a lot of places. The epidermis has got a lot of flat layers of skin cells. Okay, stratified squamous. That'll give you protection. Stratified anything. Okay. Cuboidal is next. Cuboidal or cube shapes. We can have simple cuboidal, so one layer of cubes. Your glands, so like your sweat glands and your oil glands, your tear glands, your earwax glands, okay, the glands of your body are going to be lined or formed by epithelium. So like if you think of like the skin, the surface of the skin, the epidermis, well, if we have a little hair follicle or a little sweat gland or oil gland, the surface just kind of dips down to where that pool is and it keeps going. So the lining of the gland is going to have its own kind of the lining. A gland is going to have its own lining or covering, just like your skin. We usually see simple cuboidal. It's going to be glands. Glands are like little containers, like little jugs. Okay? And each of those little containers or jugs has a lining, and it's usually cuboidal. Simple cuboidal. Stratified cuboidal, we see that in a few of your organs. I think maybe the spleen might have some of that. But yeah, many layers of cubes. We see that in just a couple of spots, not too many locations. And then columnar, and again, I have some images or illustrations on these specifically. Columnar are column shaped, and generally these are only simple. We usually just have simple columnar. Stratified columnar is kind of, we don't really see that much. But we see that pseudo stratified columnar, which I'll show you that shortly. And again, the surface specialization, we could say ciliated stratified squamous tissue, keratinized stratified squamous. So anyway, we can use even more terminology to describe epithelium. Is it ciliated? Does it have cilia on the surface, right? Is it keratinized? All right, so anyway, simple squamous. I'll go through these relatively quickly. Again, simple squamous, one flat layer, okay, flat and smooth. Okay, and again, we'd probably find this, it says mesothelium and endothelium. Okay, we're not worried about these terms right now. They're just linings. Endothelium. So again, we generally see this in the lungs or the alveoli. Okay, simple cuboidal, secretion and absorption. Okay, secretion and absorption. So we might find simple cuboidal in the lining of the small intestines. And again, we'll oftentimes see glands. Glands secrete. Okay. Glands secrete whatever it is. They secrete oil or they secrete sweat, wax. Those are what we call exocrine glands. I said glands, but I should differentiate. We do have two types of glands. Exocrine are the ones I've been talking about. Those are the ones we're dealing with now. We also have what are called endocrine. We have a whole system on endocrine. Endo means inside. Endocrine excretes something directly into the bloodstream, whereas exocrine excretes into a duct. And these ducts are what are lined with usually cuboidal epithelium. Okay. So endocrine, they directly secrete their material into the bloodstream. Those would be called hormones. Okay. Those are endocrine glands. Okay. So anyhow, let's see, simple columnar. Again, these are going to be taller, definitely less common. The simple ciliated columnar epithelium. We might find some of those again, you know, in the respiratory tract, maybe lining the trachea. These oftentimes have, again, more absorption and secretion. Goblet cells are oftentimes contained with simple columnar epithelium. Simple columnar epithelium, again, oftentimes found throughout the intestinal tract as well, along with simple cuboidal. These guys that are going to secrete some digestive enzymes, okay, secrete a mucus, kind of keep the lining hydrated. So goblet cells secrete mucus to what we call the apical surface. So when you say apical surface, we're talking about the apex. And by the way, most of these that are linings, they're lining a tube. We call them tracts, right? Tract, T-R-A-C-T, tract. Respiratory tract, digestive tract, urinary tract, reproductive tract. These are all tubes. Your cardiovascular tract. Okay. These are all tubes, and all these tubes are lined with epithelium. There's a term I want to mention. If I make a tube, the surface, the lining of the tube is epithelium. So the apical surface is what is associated with the inside of the tube, if you will. The whole, the space of the tube is called a lumen. L-U-M-E-N, lumen. L-U-M-E-N. The lumen of the tube is where the product is passing through. So where blood is passing through your blood vessels, that's the lumen of the blood vessel. When you put food in your mouth, technically, you know, your mouth is almost like a lumen, a big opening of a tube. And as you swallow your food, it goes into your esophagus. Okay, that's the lumen where the food is passing through. So my point is that apical surface of the epithelial cells is kind of forming the lumen of the tube. It's like taking a garden hose and looking at the garden hose, it looks just like negative space, right? It's just space. That space is called the lumen. All right, stratified squamous, again, multiple layers of flat cells. This is going to really help with protecting underlying tissue. Areas that we find a lot of friction, a lot of potential for stressors, where there might be a lot of traffic. So that outer layer, again, gets continually worn down and shed. So notice the bottom or the basal or the base cells, you'll notice there's a little kind of line there. That's the basement membrane. Then this kind of purple stuff way down here, that would be connective tissue then, right? Connective tissues down here. Basement membrane is here. The rest of these are going to be cells of the epithelium. And each of these little circles, by the way, represents the nucleus of a cell. You probably already knew that, but I'll put that out there too. Each of these little circles inside, those are the nuclei of the cells. So again, notice this basement layer, or the base layer, they're a little thicker, a little bubblier, a little more hydrated. And as we move up and move up toward that apical surface or the lumen, we see that they dehydrate, they get a little thinner and a little flatter. Okay. This is called keratinization, what we see going on here. So more glandular epithelium. We see the cuboidal. Some of our sweat glands, salivary glands. And again, these will be found in the ducts. So the gland is going to be deep, but it has to have an exit for its product. So as the product is exiting the gland, it has to go through what we call a duct. And that duct is typically going to be 1 to 3 layers of cuboidal, usually 2. But we do see some simple cuboidal with glands as well. So again, it's not a slam dunk. If you always associate cuboidal with glands, you're going to be good. Okay. Stratified columnar. So this is that one I said, pretty rare. We oftentimes will call it, we talk about this one, pseudo-stratified quite a bit more. We don't really see much in the way of stratified columnar epithelium. Again, we might see it in a couple of areas of respiratory, digestive, or reproductive. Typically, we'd see it maybe near some sort of duct. But yeah, I wouldn't worry too much about that one. This one, we do see big time in the respiratory tract and parts of the male reproductive, but you always want to associate, or 2 things you want to, well, 3 really. Pseudo-stratified columnar means appears stratified, but isn't, and they're columns. So you can see we don't have, if we look at normal stratified, the columns are pretty much straight up and down. With pseudo-stratified, again, we do see a little bit of alteration in some of the base cells. Okay. Now, a couple of things we see differently than any other tissue. With pseudo-stratified columnar, we typically see cilia on that apical surface. And we also oftentimes will see goblet cells too. Usually cilia and goblet cells kind of go together, because the cilia help with kind of moving particles around that might be coming into contact with the lumen. But the goblet cells secrete the mucus that kind of enables these to be a little stickier. So when particles pass through, they get stuck to the cilia that are, again, kind of sticky from the goblet cells, the mucus of the goblet cells. Okay, so definitely think in the respiratory tract. That's where we see a lot of that pseudostratified. We're almost done with epithelium. Then we'll spend a few minutes on connective, muscle, and nerve, and then we're going to jump into integumentary system. The last one we'll go through is called transitional epithelium, kind of an oddball. Transitional means it changes, right? If we transition, we're kind of changing or altering our shape. So transitional epithelium changes its shape depending on what's going on with the particular organ. We generally only see this in the urinary tract, especially the bladder. The urinary bladder is like a balloon. When a balloon is empty, it's very thick, right? The rubber of a balloon that isn't blown up right out of the package. If you get a red balloon or a purple balloon, the rubber is really thick and the color is really deep and dark. But as you blow that balloon up or fill the balloon up, it becomes thinner, right? The layers become thinner. The balloon, even the color changes a little bit. So that's what your bladder is doing. Your bladder, when it's empty, is very thick epithelium. Okay, so you may have like a base layer, maybe of cuboidals, and then a superficial layer of squamous. It allows it, as it fills or distends, it allows for more flexibility. So when we have a transitioning or changing type of situation, such as a bladder filling or emptying, we would see transitional epithelium. Pardon me. All right, cells or glands, moving on to glands. Again, I'm not going to talk too terribly much about these. I've mentioned them a little bit, but these are specialized cells that produce and discharge a secretion. And again, we classify glands primarily as being, are they endocrine or exocrine? Endocrine has to do with a secretion that's going directly into the bloodstream. So endocrine goes into the bloodstream. Exocrine secretes into a duct of some sort. Exocrine secretes into a duct. The other thing about an exocrine, it secretes into a duct. It's primarily going to be affecting its area, the local, wherever the exocrine gland is, it's affecting the area it's located. Whereas an endocrine gland is dumping its hormone into the bloodstream, well, that could go anywhere in the body. So endocrine secretions we think of like hormones, and hormones travel in the bloodstream throughout the body. So those are the primary differences or the primary types of glands that we see. It's either an exocrine or endocrine. And we spend an entire week just on the endocrine system. So we'll get very familiar with endocrine and its hormones that it produces. So again, it shows the difference in exocrine gland. You can see this is epithelium up here. The epithelium comes down and you can see it's kind of cuboidal, maybe a little, starts to get a little columnar maybe, but yeah, primarily cuboidal. It's got a duct. So it's just going to secrete its material right out of the duct to the surface. Okay. Whereas an endocrine is deep and usually found then in the connective tissue region. And we would see this is a lymph node here. we can see it's got the gland, the secretory cells, and then some sort of blood supply kind of embedded within it. Okay, so endocrine dumps directly into the bloodstream. We're going to keep moving. I'm not going to get too into the details of specific types of exocrine. Oh, yes, so apicrine, I'll mention that really quick. Apicrine glands typically, these are sweat glands. Apicrine, typically we'd find on the head and the armpits. Types of secretions, just again briefly, because I want to move into connective tissue. Serous, it's like a serum. When you see serous secretion or a serous membrane, it's dealing with watery, very thin liquid. Okay. That helps, like when we looked at you guys looked last week a little bit at visceral versus parietal, and in between the visceral layer is up against the organ, surrounds the organ, and viscera means guts or organ. So visceral anything is going to relate to the organ. So a visceral layer or membrane is going to go right around the organ, right up against it. Then you're going to have a little cavity or a little space. And then you're going to have the parietal layer is going to be more superficial. So visceral is deep against the organ, parietal is up over top of that, and then there's a space in between. Oftentimes that space is going to have a fluid in it. Okay? And that fluid is typically what we call serous. In fact, those are called serous membranes that surround different organs. And what that does, for instance, like the heart or the pericardium or the pleura, the lungs, they have a lot of action. So there's a lot of friction created by the constant contraction. So if I put that muscle inside of a double layered with a water lining, that's going to reduce some of the heat and friction. So fluid is normally around the heart, but it's regulated and it's contained in a small kind of a cavity between these two layers. A more sedentary lifestyle for animals, for humans, for anything, any creature, the more sedentary that animal is, the less activity the heart is getting, and so the more likely that we could get some fluid buildup. So it's good to have some activity and to not be entirely sedentary. That doesn't mean either that we're trying to get you get an animal that's used to, or yourselves, you're used to being paced and doing certain things, and then saying, oh, now we're going to run and prep for marathons. We haven't been doing anything, and now we're going to hit the ground running. That's not healthy either. That can trigger all kinds of problems with inflammation around the heart as well, and fluid balance issues. Okay. So anyway, and mucus is just that, thick, more viscous. We see mucus more in the guts, and then maybe like the upper respiratory tract, and a little bit in the lower respiratory tract, but the cilia help to kind of keep that mucus moving. All right, so the big chunk is done. We just have to do a little bit with connective, muscle, and nerve, and then I want to talk briefly about the integumentary system. Looks like we've still got about 45 minutes or so we're right on task. All right, so connective tissue is just that. It's what connects or links that epithelial covering or lining with the musculature that is deep. Okay, so connective tissue provides the vascularization. So it's responsible for a lot of the metabolic. By the way, when you see metabolic or metabolism, it just means chemical reaction. And we have two main types. There are more, but the two that we work with primarily when it comes to metabolism are what we call catabolism with a T, catabolism, and anabolism. What we call them is synthesis or decomposition. So metabolism simply means we're putting things together chemically and building something, or we're tearing something down and breaking it into smaller parts. So like I eat a piece of steak, the body metabolizes it into or breaks it down into amino acids. And then my cells, through protein synthesis, are going to synthesize or put back together or metabolize the amino acids back into protein. So metabolism goes both ways. Break it down, build it up. That's the entire life of your cells. Yeah. Your cell, oh, by the way, I mentioned this and you'll hear me talk about it in the video a little bit. Just as a side note, when we're talking about organ disease or organ inflammation, organ illness, we're talking about the cells of that organ at the cellular levels where the problem is. Pancreas isn't sick when you have diabetes. It's the cells that deal with, it's the Langerhans cells and these islet cells. These are the cells that are having problems. Thus, the whole organ has a problem. So we have to start at the cellular level. That's why we do diagnostics. That's why we draw blood, we pee in cups, we breathe in tubes, we do different things to get some sort of diagnostics on maybe what's going on the cellular level. We have to address that. All right, so anyway, connective tissue. Insulator, it's, again, highly vascularized, a lot of blood supply in connective tissue, a lot of nerve supply as well. It's the most abundant of the tissues, and there are a couple of main features. For connective tissue, just think of it as being cells inside of or within what they call a matrix. The matrix is what we call ground substance, which is going to be, it's going to have some water and some electrolytes and some protein. And then we have extracellular fibers. So we're going to have some keratin or collagen fibers, elastin fibers. We're going to have different protein fibers. Okay. But yeah, cells within a matrix. The matrix could be primarily fluid. It could be just primarily fluid. Usually it's not. Usually there's some globular proteins and some other solid material within the matrix. So connective tissue, cells within a matrix. Remember, epithelial tissue is just cells compacted together. There's no matrix. So that's the difference. Epithelium is cells packed, connected onto one another. Connected is cells swimming or floating in a matrix. So blood is considered connective tissue because it's red blood cells, white blood cells, and platelets floating, suspended within plasma, which is the matrix. Bone is connective tissue. We'll see next week. You've got a bunch of little osteocytes or bone cells surrounded by or embedded within a matrix. Not as liquidy, right? Bone, the matrix of bone is more ossified. It's got more mineral content. Primarily what? What makes up bone? Supposed to drink lots of milk, build strong bones. Like calcium, right? Calcium and phosphorus are the big matrix components of bone. But we'll see that next week. Fat, cartilage, bone. Okay. So again, that ground substance, that's the medium through which the cells exchange nutrients and waste with the blood. It's basically the extracellular fluid or the extracellular space. You get some funny words: amorphous, homogeneous material. you don't need to know that's a little heavy. So round substance. That's just simply the medium in which the cells are kind of suspended or living with it. Okay, and then we have some fibers. There's always going to be some fibers in that matrix. Primarily, we're going to have collagen fibers. You guys have all heard of that at some point, I'd imagine. Collagen fibers. Collagen provides strength and durability. And then we have what are called reticular fibers. These aren't as strong. They're a little thinner, more delicate. So we'd find these in some of your real solid organs. It says highly cellular organs. Organs are either hollow or solid, right? Like your digestive tract. That's pretty much what we call a hollow organ. Whereas like your liver, your spleen, bone marrow, these are solid organs, densely containing, densely compacted cells. Okay, so reticular fibers. Collagen's thicker, provides durability and support. And then lastly are elastic fibers. Just like they sound, these are going to provide flexibility. They're comprised of something called elastin. Yes, places where we need to have flexibility or stretchability. So vocal cords, lungs, skin, blood vessel walls, a lot more elasticity in those areas. We'll keep moving. A couple different types of connective tissue. Connective tissue is always abbreviated CT, by the way, C period T period. You have to keep writing it out. We have two types, well, we have multiple types, but two main categories, loose or dense connective tissue. The loose and dense adjectives relate to fiber amount. So we just looked at, you can have collagen fibers, reticular fibers, and elastic fibers. So there's a lot of protein fibers involved within that matrix of connective tissue. So what you end up with is how much of it is there. If there isn't a lot of it, we call it loose connective tissue. If there's a lot of fibers, we call it dense connective tissue. And then from there, we can discuss the fiber arrangement. So like dense connective tissue, we can say, well, it's dense, so there's a lot of it, a lot of fibers. How are the fibers arranged? Are they arranged in a parallel fashion? We would say that's regular. Dense, regular would be a lot of fibers, nice and neatly arranged. Maybe see that with like tendons or ligaments. Dense, irregular is a lot of fibers, but there's no pattern of arrangement. They're just kind of scattered and clumped together. We'd see those where we have movements that go in multiple different planes or directions. Whereas regular is going to give you more like uniplanar or uniaxial movements. It's like a series of strings parallel with one another. So you pull on all those strings, you're going to get the same action. Irregular, they're going to be scattered all over the place. And then elastic tissue, again, we're going to find a lot of fibers because it's dense, but they're going to primarily be elastin or elastic fibers. Again, we'd find that wherever we have areas that we need more flexibility. And then going up to loose, and again, I have a few illustrations of these: areolar, adipose, and reticular. So we'll look at what those all mean in a second. Here's areolar, loose areolar connective tissue. Again, it's showing you in this drawing, we've just got We have some fibers, they're kind of spread all over the place. It's just kind of loose in here. And again, we would primarily see what are called fibroblasts, which again are going to produce primarily collagen fibers. So we're going to see quite a few little collagen fibers within that areolar tissue. All of our mucous membranes are going to be areolar. If we get excess fluid in areolar tissue, because it is loose, there's room for excess fluid buildup. So we would see this maybe in the dermis of the skin, right? Areolar tissue. If we get excess fluid, that can cause edema to build up. So it's giving you a pathology associated with areolar tissue. And edema, if we've got edema in tissues, especially if we see it in the appendages, for instance, down at the ankles or in the feet, that's oftentimes an indicator of something problematic, maybe with the kidneys or the lungs. Because kidneys and lungs do a lot with fluid maintenance, fluid balance, pH balance. Okay. So anyway, areolar tissue. Random fibers suspended in a thick ground substance. Adipose just means fat. So we have a couple different types of fat tissue. We have white and brown adipose. Adipose is highly vascularized. These are storage units for energy. So these adipose cells are very flexible. They can store a lot of energy. They also provide some insulation for temperature and certainly absorption of shock. And then reticular tissue, we see a lot of thin, loosely arranged fibers forming a stroma. We primarily see reticular tissue in the spleen, lymph nodes, and bone marrow. So one of the ways to identify, so blood is, the spleen does a lot with blood, red blood cells, lymph nodes certainly as well, and obviously bone marrow deals with blood. But generally we see reticular tissue, if we look at it under the microscope, we'd see a lot of red blood cells kind of mixed in with it. So that's one of the indicators. Notice we don't see really any red blood cells with adipose. We might have a couple with areolar, but notice we have tons of blood cells. That's an indicator that we're looking at reticular tissue. And then the dense fibrous, we have regular, dense regular. Again, the collagen. So dense means we've got a lot of fibers. Regular simply means that they're nice and neatly arranged. So that's what we have going on here. And again, we'd see this with like tendons and ligaments. Tendons attach to muscles, which attach to bones. So tendons usually, again, are going to help to provide some movement at a joint. And then ligaments are going to be connecting a bone and a bone. So we'll look at these a little bit more next week when we study the musculoskeletal system. So this is kind of your first real introduction, I suppose, to the musculoskeletal. We've talked a little bit about some bone cells, but tendons and ligaments are major connective tissue aspects of the musculoskeletal system. Tendons are going to attach a muscle to a bone. Tendons attach muscle to bone. So when a muscle shortens or contracts, it's going to pull on the bone. Well, it's going to pull on the tendon, which then pulls on the bone. So tendons enable or kind of accommodate movement. And again, most muscles kind of move in a single plane. So we see, again, that regular arrangement of the fibers. Ligaments are going to connect a bone and a bone. So ligaments are going to be found kind of around a joint. The definition of a joint is where a bone and another bone meet. Okay? So around that joint, or where those two bones meet, we're going to find ligaments. And ligaments are going to provide durability. They don't provide much in the way of flexibility, They provide stability and durability. So again, we're going to see the fiber arrangement be very parallel. So every, like the tip of your finger and the middle part of your finger, that is a joint. You have a bone and a bone. They're two different bones. One bone here, one bone here. You have ligaments connected out here and out here. Kind of keep that joint from wobbling too much. Then on the underside, you're going to have the tendons that attach to the muscles of your forearm that allow for the movement. Then you have some of the best. These are tendons that you can see back here. You pull on the tendon, you get movement. Ligaments are usually lateral and medial. It's like in your knees, you have lateral and medial ligaments. Your elbows have lateral and medial ligaments, all your fingers. We'll get into that in more detail next week. Oh, and there's that term fascia. Fascia, I've mentioned that before as well. So next, we're going to see fascia next week. Fascia is what covers muscle. Fascia is also what connects that subcutaneous fat, what we call the hypodermis, with the muscle. So you got a muscle, and then you got the fat layer. Well, fascia is in between it. And then in between each muscle is fascia. You can kind of see on these bone models or these muscle models, a lot of the, like the gray parts, those are going to be like tendons. All this gray stuff, those are muscle attachment sites. Oh, yeah. I guess maybe like some of this white stuff in between would maybe be fascia. Now I thought one of these, you can't really tell from where you're sitting, but this side is brighter than this side. I don't think you can tell from there. But this is covered in fascia. This is not. We've removed the fascia. So next week when we start dissecting, you're going to be dealing with a lot of fascia. Fascia is kind of what separates or divides X muscle from Y muscle. When I dissect, it just looks like a bunch of gray kind of fibrous blobs together. There's some whitish kind of lines in between. And that's fascia. We can take our probe or a scalpel and get those and separate the muscles out. So you actually see this is the pectoralis major, this is the deltoid, this is the bicep, this is the tricep. On the animal, it just looks at it, on us, it just looks like all one muscle. The fascia. Fascia is what oftentimes causes limited range of motion problems with fascia. Fascia is where micro tears occur if you kind of overstretch or a muscle. So those micro tears get fixed with adhesions. And if I put adhesions directly under the skin with that fascia, it can limit range of motion. So a lot of physical therapy and a lot of occupational therapy deals with trying to get rid of excess fascial scarring. Any surgical procedure that you'll do or participate in as vet techs, you're actually cutting through epidermis, then dermis, then subcutaneous or hypodermis, and then fascia, and then the muscle. And fascia doesn't heal real well. It's kind of, like I say, it piles a lot of tissue, and you end up with a lot of scarring that's left behind, especially when you see scars that are kind of grooved, as opposed to like some scarring, like if you get a good cut, you know, and it gets stitched properly, you'll have a scar, but it's level with the surface. You have some scars that kind of, you can feel like a depression. That's because of the fascia underneath didn't get sutured properly, and so it just kind of piled up, and so you're left with kind of a groove where the skin at the bottom was sutured or where it healed. Okay? So if we suture, we want to suture layer by layer by layer, not just grab a whole bunch of layers and suture them all together, unless it doesn't matter, right? Some animals, it doesn't matter what kind of scar is left, but it can inhibit range of motion for anybody, even an animal. Cause discomfort, it cause blood supply issues. All right, so what else are we doing? Elastic, again, a lot of flexibility. Cartilage, so other types of connective tissue I mentioned earlier. Blood, we have cells in plasma. Bone, which we'll look at next week, as well as cartilage. We'll see 3 different types of cartilage next week when we study bone. Hyaline, elastic, and fibrocartilage. Again, cells in a matrix. Cartilage is gristle. Very chewy. It's avascular, no vascularization with cartilage. That's why it's very difficult to heal when cartilage is damaged. No nerves, no vascularization. A lot of cartilage is oftentimes found between joints, depending on which cartilage we're talking about. But the cartilage that's found between joints is there as like a cushion. Helps with shock absorption. Your intervertebral discs, the little discs in between your vertebrae, those are cartilage, cartilaginous disc. They call it fibrocartilage. And that's there for shock absorption and support. When someone or something injures or damages cartilage, usually doesn't heal. So we have to, so oftentimes we'll remove it or we'll buff it. Well, they call it clean it up. Some animals have to get their hips cleaned up or their knees cleaned up because they have cartilaginous fragments that all affect the range of motion because the joints are in a capsule. So that capsule inside the joint isn't getting any blood, but there's cartilage inside of that joint. If it gets torn a little bit, it just stays in that capsule as like a floating chunk of cartilage or maybe a flap that tore off. So every time the animal or you bend your knee or bend a joint, it gets caught or it catches. So cartilage won't heal itself. So that usually requires arthroscopic type surgery. Okay, hyaline cartilage, the most common type. And again, notice it's cells contained in a matrix. Cells in a matrix. That makes it connective tissue. Elastic cartilage. You see some elastic fibers in between the cells. And then fibrocartilage has a lot of fibers. So it kind of goes from least fibrous to most fibrous. So hyaline cartilage is least fibrous. You can see it's this under the microscope, like in real color, it looks almost like opalescent or almost translucent with like kind of that rainbowy effect to it. Hyaline cartilage. Call it milk glass appearance. You'd have to be like an antique dealer to even know what milk glass is. But anyway, opalescent, maybe you've heard of that, kind of like a shell, the inside of an oyster shell. It's kind of a rainbowy look to it, like a sheen. That's what hyaline cartilage looks like. Elastic has fibers, and then fibrocartilage is full of fibers. So this is going to be the vertebrae of the spine. This is the pelvic joint, called the pubic symphysis, and then the knee joint. Bone, we'll get into bone more next week. You got to keep her moving. Same with blood. We do a whole week on blood. Again, cells in a matrix. These are red blood cells, white blood cells, and platelets floating in plasma. About 55% of your blood is plasma, and about 45% is cellular. Okay, membranes, I think we touched on these a little bit, or you did maybe last week. But we talked a little bit about linings, the visceral and the parietal membranes. But we have mucus, serous, cutaneous, and synovial membranes. Again, mucous membranes are primarily going to be lined with some sort of columnar epithelium with goblet cells. We do see some stratified squamous as well, especially in the nasal cavity. just because it's such high traffic area. Okay, so mucosal layer. Basically, the respiratory digestive tracts are covered with mucosal membranes. The mucus, remember, is kind of a thicker fluid. Oftentimes aids with movement of product, whatever that might be. And just deep to the mucosal membrane is called the submucosa. So again, that's just threw that in there because we will see this later on. You know, we talk about this now, but like mucosal membranes, we probably won't see these again for like 3 or 4 weeks. Mucus. And serous or serosa, that oftentimes is what we find in those cavities of the walls, you know, between the visceral and parietal layers is the cavity. That's where the serosa is oftentimes found. And again, it helps with kind of reducing friction, helps with hydration. So in the lungs, we've got pleural fluid that surrounds the lungs, peritoneal fluid in the abdomen, and then pericardial fluid around the heart. All right, 2 more members, the cutaneous and synovial. So cutaneous, whenever you hear that, it always relates to the skin, right? Cutaneous. Think of like your cuticles, maybe. But yeah, cutaneous equals the skin. So if we say subcutaneous, we're talking about below the skin. The fatty layer below the skin is called the subcutaneous fat layer. Cutaneous. And then synovial membranes are the last form of or type of membrane. They don't have an epithelium. These are typically found around your movable joints. So if it's a movable joint, it's good. I mentioned just a few minutes ago that they have a capsule around them, a joint capsule that's lined with a synovial membrane. And the fluid inside of these joints is called synovial fluid. Very viscous, very slick. It's what the Tin Man was lacking. If you watch Wizard of Oz, he didn't have any synovial fluid, so he couldn't move around very well. So let's see, we've got a little bit of muscle to go. That's just 3 things right here: skeletal, smooth, cardiac. That's really all you need to know right now about muscles. Next week, we'll get into detail about skeletal muscle. The other 3 things, so there are 2 things with muscles regarding classification. They're either movable, like voluntarily, right? So we call it voluntary movement, or involuntary. So skeletal muscle is certainly voluntary. And then smooth and cardiac are involuntary muscles. That means we don't control them consciously anyway. There are ways you can, through coping mechanisms, meditation, prayer, relaxation techniques, right? Focus. A lot of athletes and a lot of performers of whatever go through breathing and calming. They call it the Alexander technique of posture and kind of slowing their heart rate down a little bit. So you do have some control of your heartbeat, but you don't have control of it beating or not beating. It's always going to beat until we're dead. Okay? Involuntarily, we don't have to tell it to. We wouldn't get anything done, right? Our heart beats probably about once a second. If you had to tell your heart to beat every second, you'd be screwed. When would you sleep? You know, and if you didn't tell it to beat, it would stop beating. Well, you can't have that. So it's involuntary. Same with smooth muscle. From mouth to toilet is probably 25 feet, maybe 30, depending on the person. 25 to 30 feet from mouth to toilet. I don't have to spend 1 second of the day telling my intestines to move that food to the toilet. It just does it. That's smooth muscle. Okay? Your reproductive, your urinary tracts, those are smooth muscle. Remember, if it's a tube in the body, it's smooth muscle at the deep part. It's lined with epithelium. Then we have connective after that, and then finally the smooth muscle. So respiratory tract, digestive tract, urinary tract, reproductive tract, your cardiovascular system. Those are all tubes, and they're all smooth muscle. Okay. So voluntary. The other term is it's either striated or not. So striated means stripes. So voluntary striated. That means I have control over the movement and it has stripes. Okay. Smooth muscle is none of the above. No stripes, that's why it's smooth, and no control. So involuntary, non-striated. It's smooth. Peristalsis is the move. So what are my skeletal muscles? They're just contracting, relaxing. That's it. Contract. So a muscle either contracts or it relaxes. How do I get smooth muscle? It's a tube. Smooth muscle works through squeezing. We call it peristalsis. I don't know if you can see that at the bottom there. Peri means around, and stalsis has to do with squeezing around. All right, and then finally, cardiac is your heart muscle. That's striated, just like skeletal, but it's involuntary, like smooth. Nerves, we spent a whole week on that. Nervous tissue, simply neurons and glial cells. Okay, so remember, tissues are cells. So the cell types for nervous tissue are called neurons and neuroglial cells. Oh, for muscle, the kind of the combining form or root is myo, M-Y-O. So a myocyte would be like a muscle cell. We'll see words like, we call them muscle fiber, to be specific. You guys have probably heard that, right? Muscle fiber. A muscle fiber is the muscle cell. A neuron is the nerve cell. We also have glial cells. Glial means glue. This is like the white matter that surrounds neurons. Glioblastoma is a brain cancer of the glial cells. Neurons don't go through mitosis, but glial cells do. So the good news is you can't get nerve cancer. Bad news is you can get glial cancer. Anyway. You can't get heart cancer either. You ever heard of that? Heart cancer? Because it doesn't exist. Because heart cells don't go through mitosis. That's good. Heart deals with a lot of other stuff though, right? Got tiny blood vessels, arteries that get clogged real easily, and it won't beat right. So anyway, neurons and glial cells. We're not going to get into tissue, wound healing, or any of that at the moment. All right, lastly, I just want to spend a couple minutes on integument, and we'll move on with life. And then next week, we'll start cutting through the integumentary system. Now, these first couple of items we've talked about a little bit as far as tissues go, the epidermis versus the dermis. So again, the epidermis of the skin is what we can see. So that's epithelium. Epidermis is epithelium. The dermis is then the connective tissue. And then we have the hypodermis or the subcutaneous layer. That's primarily fat or lipid. We know adipose is a type of connective tissue. And then deep to the hypodermis would be then the muscle tissue, the muscle layers. Those are your three layers: epidermis, dermis, and hypodermis. Technically, the hypodermis is not part of the skin, but we're not going to worry about that. It's under the skin. It's called the subcutaneous. You see, that's where a lot of fat would be. Now, epidermis, being that it's epithelium, it's primarily made of keratin cells. Okay? So keratin cells are probably 85, maybe even 90% of your epidermis is keratin. Okay. Then you've got maybe about 6 to 8% are melanocytes. These produce the pigment of our skin. And then lastly are Langerhans and Merkel cells. These are probably less than 1% of your epidermal cells are these guys. Merkel cells, again, they're going to link up with the nerve endings that come up through the dermis. And Langerhans cells help with the immune system. Okay, so those are the epidermis cells. The layers of the epidermis, and again, the video in Padlet for week 2 goes through quite a bit of what we're talking about and what we've talked about today and what we're talking about. So if you've watched that, then some of this is familiar to you already. If not, then yeah, there you go. So stratum means layer. So for us, we're going from superficial to deep. So the top layer is called the corneum. So that's the layer we see. So the layer of epidermis that we see is called stratum corneum. Then we have lucidum, granulosum, and spinosum in the middle. Lucidum is only in thicker skin, so be like the palms of the feet, and the bottom, or palms of the feet, palms of the hands, bottom of the feet. We would find thick, the lucidum. Granulosum is the granular layer. This would be the waterproof layer. So this enables us to not take on water when we get in the swimming pool or take a shower. The skin doesn't, the skin's waterproof due to that. And then lastly, the spiny layer. This spies kind of like keeps an eye on, pardon me, keeps an eye on the base layer. The base layer is also known as basal or germinativum. Germinate has to do with reproduction. So again, that's this basement layer, stratum basal, that's where all the mitosis is occurring. And then it moves up. The spinosum or the spy layer is basically kind of keeping an eye on the germination of these basal cells. So anyway, the basement layer all the way up to the top layer, basal to corneum. So know those layers. And then the dermis, again, that's the connective tissue. It's dense, irregular connective tissue. And again, we find the 3 primary fibers: collagen, elastic, and reticular fibers. It's also where we find everything else: your hair follicles, your sweat glands, your oil glands, nerve supply, lymphatic supply, blood supply. Basically, all the stuff that we need for our skin is contained in the dermis. We have two layers, the papillary and reticular. The papillary is going to be the most superficial layer. The papillary layer of the dermis is what connects with the epidermis. Papillae are bumps. Papilla is a bump. So we've got these little bumps at the top of the dermis, and those plug into the basement membrane of the epidermis. And then the reticular dermis, again, that's where we've seen the word reticular connective tissue. Reticular just means we've got all kinds of fibers around, all kinds of action. Okay, so yeah, I mean, the reticular layer, that's where everything is. About 80% of the dermis material is in that layer. And then lastly is the hypodermis. That's a thick layer, primarily of fat. We do see some nerve there as well. So for pigmentation, we have melanocytes. Sorry, I'm zipping through this, but we're just about at the end of the road, I think. Yeah, we have what, 8 minutes maybe left. Yeah. So, some special features of the integument: pigmentation, paw pads, planum nasale, ergots and chestnuts, and cutaneous pouches and sheath. Okay, so pigmentation, for one, we have what are called melanocytes. The absence of melanocytes is what leads to what we call albinism. So you'll see some, you've seen rabbits, mice, rats, even humans that don't have melanocyte activity. So they have what's called albinism. Their eyes are going to be red, right? All white bunny rabbit with the bright red eyes, they lack melanocytes. Controlled by the hypophysis, well, that's going to be that's pituitary region. Okay, so the pituitary in the brain, the anterior and posterior pituitary, we'll look at in about 2-3 weeks. Okay, paw pads, the tough, thick layers of fat and connective tissue, they have what are called conical papillae. Remember, papillae means bump. So they have little coney type bumps that cover the entire pad. And again, all 5 epidermal layers, just like us, you have that lucidum. Lucidum is only with thick skin. So you're going to have, most skin only has 4 layers, but again, paw pads, bottoms of the feet, they're going to have all 5 epidermal layers. They're generally also going to be pigmented. And we have a couple different types. We have the carpal pad, the metacarpal pad, and the digits. Carpal means wrist. Metacarpal has to do with the hand. So for us, this would be our carpal. This would be our metacarpal. And then this would be our digits. Okay. So same thing going on with puppies and pooches and kittens and cats and all that good stuff. And then the nose, right? Top of the nose in cats, pigs, sheep, dogs, it looks a little different, certainly than ours. If you look up close, there's a lot of little tiny grooves, and these are called plaques. Again, usually it's going to be pigmented. Aglandular, except in sheep, cows, and pigs. In dogs, you're going to have 3 epidermal layers. The corneum which is usually a little thicker, is going to be thinner, and it's going to, again, it's divided up in these little grooves. Different plaques and what they call plaques and grooves. Ergots and chestnuts. Again, those are those kind of dark structures you'll see. Chestnuts. Right around what would be kind of considered their wrist and ankle areas. And then down near kind of like the palms of their hooves, kind of toward the back there. In the caudal hairs of the fetlock, we will find those ergots. And sheep have what are called cutaneous pouches. fine hair with sebaceous and oil glands. Sebaceous means oil, so it's kind of redundant. Sebaceous and oil glands, we just call them sebaceous glands. So they have infraorbital. Infra means below the eye, below, and orbital means the eye. Interdigital means between the digits, and then inguinal means in the groin. So they're going to have some extra pouches that spritz oil. And again, that helps certainly with keeping their, I don't know if you've ever pet a sheep or been around sheep, but their skin's really oily, or their fleece is pretty oily. They have extra pouches for oil secretions. We'll dissect sheep GI tract this semester, so that'll be fun. The whole thing, mouth to anus of the sheep. We find different things inside their stomachs. Anyway, that's it. We got a little, there's some hair and nail stuff too. You can go through, but for the most part, we don't really do a heck of a lot with that. Primary hairs, secondary hairs, and tactile hairs. Tactile hairs are again, just like they sound, tactile to do with sensation of touching. So you know that many animals with their whiskers have little feelers or probes, and that's how they're able to get around and sense different things. There's introducing your first muscle of the semester. Your first muscle is called the erector pili. Palo has to do with hair. Erector has to do with standing on end, being erect. So the erector pili gets the hair to stand on end. So we see this during fear, primarily. Also see it with temperature issues, right? Temperature changes, goosebumps. These cause goosebumps as well. And it's the sympathetic nervous system. So here's another, so you're getting your first muscle and your first real dig at the nervous system. Sympathetic nervous system is your body's stress nervous system. Your body's response to stressors is what triggers the sympathetic nervous system. We call it fight or flight. There's a third F word called freeze. Some response to stress isn't to fight it, isn't to run from it. Sometimes it's frozen. So anyway, so the hair standing on end is a normal response. Glands, we've talked a little bit about these already, right? Sebaceous glands, sebum, these secrete oil. Sweat glands, again, we have ecrine and apocrine. Tail glands and anal sacs. We'll go right to those. Tail glands, most dogs and cats at the base of their tail are going to have a little bit of a gland spot there, and this isn't their anus. And this is a right near there, but it's a little further up on the tail, the dorsal base. Okay. Cats especially, right? You'll watch a cat walk around a corner and they'll like they'll kind of swirl the tail around the corner, right? That they're marking that. And then underneath, so that's on the dorsal aspect. The dorsal, top. And underneath, right next door to their anus, we're going to have a couple of little glands that secrete, called anal sacs. They secrete kind of a funky smell, especially if they haven't been expressed for a while. But typically, with the dog, when the dog poops or the cat poops, those anal sacs get kind of stimulated. So part of the stench that goes with their poop is because of the anal sacs. But even if they're not pooping, if they're frightened, scared, territorial, any tweak outs like that, that could cause the anal sacs to express. And depending on the animal, dogs, it oftentimes smells kind of like tuna. It's also, anal glands can also be used for finding a mate, attract, it's like that, pheromones, if you will. Skunks probably are the most well-known anal glands on the planet, right? They've got really pungent anal glands. And yeah, they can get infected. Certain dog breeds are a little more susceptible. I think they're finding that, like, tails that have been cropped or snipped, sometimes those dogs have a little more, like boxers have, and like Boston Terriers, they sometimes have anal gland issues more than others. But anyway, so yeah, as a vet tech, you might get to express an animal's anal glands. Usually we try to tell the patient or the owner of the patient that they could potentially try to do that at home. A warm compress. But it could be, if it's a recurring issue, then sometimes they'll remove them. We'll remove them. You're kind of eliminating like a part of that dog, right, or that cat. It's a big deal. Anyway, claws and dew claws. And again, dew claws are kind of what they call a vestigial remnant or evolutionary remnant of digits. So dogs oftentimes will have a dew claw. A lot of places they'll lop off that dew claw. So, but anyway. A hoof is also known as an ungula. So all hooved animals are called ungulates. And in the horse, we've got 5 different types of corium. That's why a farrier, horseshoers, if you've ever watched that done or been around that, I mean, it's pretty intense. It's pretty neat. It's fascinating to see all the goop and gunk and what it's a major aspect of horse health. Major aspect. So, yeah, a lot of calls that we've gotten with horse problems are because they haven't been taken care of. There's the feet. So yeah, they need their nails did and their hair done. All right. So anyway, you guys can go through this a little bit on your own. I've got a couple questions in there that go through them. So other than that, I think I'm pretty well worn down, and you probably are too. There's a lot of info crammed in there, but we made it. You guys have questions? Okay, good. Email me if you do. If you ever have any issues, just let me know.